I'd like to thank you for joining me in this short discussion regarding temperature uniformity testing, or what most refer to as temperature mapping. In the past few months, I've received multiple questions focused on how to successfully map a controlled temperature instrument like a freezer. The purpose of this video is not to explore the complexities and details of temperature mapping, but rather to talk about the basics of mapping and to address some issues that might affect your mapping results. I'll be sharing a few simple tips that you may be able to use to ensure that the results you acquire through testing truly reflect the temperature condition of the freezer. By following these simple tips, you'll be closer to collecting correct and accurate results with a robust and repeatable protocol. Some of these tips may seem a bit too obvious for some, but don't be fooled. Failure to consider any of these tips may dramatically impact your test results and affect the potential safety of your samples. So let's get started. So the first tip is to be sure that you order the correct freezer. Now this can be done by answering a few questions that are important to your specific application. First of all, what regulations do you need to satisfy? A few common references are the FDA, ICH-Q7, and the United States Pharmacopoeia. Have you benchmarked organizations and societies to see what is common practice for your type of application? There are several best practice guides available that might be able to help. There are the International Society of Pharmaceutical Engineering and the International Society for Biological and Environmental Repositories, just to name a couple. Also, and this is very important, what is your qualification performance test criteria? You need to get specific here, as the test acceptance criteria will determine which freezer can be used. Depending on your needs, the United States Pharmacopoeia might be a good option for this information. The second tip is to follow good documentation practices. It is critical that you have a system in place that drives your efforts that would include the decision-making process and purchase of the freezer through and including the final qualification. A good practice is to follow the commonly used DQ, IQ, OQ, PQ common practice. This will ensure that you have documents that will allow traceability throughout all parts of the qualification and can be repeated easily if needed. Be sure to document as you go so nothing will be forgotten. Previously, I mentioned a DQ. This is important as it provides a checklist of facility requirements your new freezer will need. Your DQ should include electrical requirements, ambient temperature, and heat load. You don't want to install 10 freezers in a room that will end up getting too hot, and it might activate the overheat safety feature. One of the most common mistakes I've seen is planning for the size of the freezer. Will it fit through all of the doors from the loading dock to the laboratory? All of this information can be found on the technical data sheet. So what does all of this have to do with mapping? A lot. I would guess that the majority of the mapping issues regarding new freezers is that the wrong freezer was ordered and would not pass the qualification tests. It's like when you have a painting project. It usually takes longer to prepare the surface for the painting than it does to actually do the painting. Proper planning and purchasing is a must. Now to the actual mapping exercise. Take a look at these probe locations. Can you find anything wrong here? If you look closely, the probes are not uniformly placed, and there are a few that are touching solid surfaces like the wall of the freezer or a shelf. This setup will most likely produce incorrect results and will possibly fail the test. It won't be a fair representation of the condition of the freezer. Be sure to consult your mapping protocol that should explain in detail where your probe should be located. Now here is one example of good probe placement. All of these probes are uniformly placed. No probes are touching the walls or laying on the shelves. They are also secured so that they will stay in place for the duration of the test. Keep in mind that this is only one way to attach probes. Do what is best for your situation. Okay, our preparation is almost complete. Once the probes are attached and the freezer is equilibrated, start your testing instrument to collect a sample of a few minutes or so to see what the temperature trend looks like. Now, if the trend is high or low, just adjust the offset or set point to center the trend on your desired temperature. Collect enough information to satisfy that you are on center. This practice is one way to maximize the performance of the freezer. It will also help to avoid unnecessary test failures and can maximize your predetermined temperature alarm limits. Let's take a look at the trend on the left. The peak value is great, but it's not centered on the desired set point. The trend on the right has been centered. This allows for less nuisance alarms when opening the door to retrieve samples. On rare occasion, you may experience a test failure. Be sure that your protocol has specific instructions on what to do in these cases. Documentation is critical, as you will need to follow your instructions closely. If you want to hedge your bet on a seamless installation and qualification experience, utilize the Thermo Fisher Scientific Service and Testing Team. They have the experience to perform your IQ, OQ, and other temperature mapping needs. A huge benefit of our service organization performing the testing activity is that if there happens to be a problem, they'll be able to address it immediately. I mean, who better to have on site than the experts that are most familiar with the product and the proper testing practices to be successful? Add to that the guarantee we offer on the validation work on Thermo Fisher units that should the customer have a failure while the instrument is under grade 1 of the warranty or qualifying agreement and deem it necessary to requalify, we'll do it at no charge. Now that we've covered the tips, let's go over what we've discussed. Create discovery questions. Know what you need before you buy. Document the activities from the beginning of the purchasing process. Request a technical data sheet to use as a reference for specific performances and physical data. Proper probe placement is critical, and you must be able to duplicate the process. Dial in the freezer. Our freezers are built to perform with a small peak variance. Take advantage of that quality and center your trend to avoid nuisance alarms and unnecessary test failures. Include specific directions in your protocol for a failure. So there you have it. A few tips that might help you avoid some of the common mistakes being made that result in unnecessary test failures. There is nothing more aggravating than failing a test only to find out that the freezer was ordered was not the correct freezer for your application, or there was a problem with the process or setup and not the freezer. Once you get a proper and robust plan in place, mapping won't be such a headache for you. You will experience a huge increase in successful tests that truly reflect the condition of the freezer, which is the whole point of the exercise. Thank you for your time.